Dear student, in the last video I discussed about the two cell organelles that are involved in endomembrane system. The other two cell organelles which are involved in endomembrane system that will be discussed in this video. So if you take the other two membrane bound cell organelles which are involved in endomembrane system, one is the lysosome, the other one is vacuole. So let us understand about the lysosome, that is what is required for the board exam, as well as I'll be taking some MCQs. But let us recollect what we have studied in the last class. So in the last video I've discussed about endoplasmic reticulum. Endoplasmic reticulum, these are flattened sac-like structure which are scattered all over the cytoplasm, usually found around the nucleus. Sac-like structures which are called cisternae. This endoplasmic reticulum, it is the largest cell organelle which divides the cytoplasm into the compartment. So you find two compartments. It has two compartments. The space that is present inside the endoplasmic reticulum, that we call it as luminal compartment, and the outside the endoplasmic reticulum, that we call it as the extraluminal compartment. And also we have discussed about the types of endoplasmic reticulum based on the presence or absence of ribosomes. If the endoplasmic reticulum is associated with the ribosome, then it is called rough endoplasmic reticulum. Endoplasmic reticulum without ribosome, that we call it as smooth endoplasmic reticulum. If it is associated with ribosome, rough endoplasmic reticulum. If we take the functioning of smooth endoplasmic reticulum, involved in detoxification as well as involved in synthesis of lipid as well as steroid hormones, particularly in animals. But rough endoplasmic reticulum involved in synthesis of protein. So whatever the protein that is synthesized, that should be properly packed. That's why the protein produced inside the endoplasmic reticulum, it will be sent to the Golgi apparatus. These are the transporting vesicles that joins with the cis phase of Golgi apparatus. Then later there will be the packaging inside the Golgi apparatus. That's why the Golgi apparatus is called packaging center of the cell. Then what are the packed material that should be sent outside? It will be released. This phase of Golgi apparatus, we call it as a trans phase. This we call it as cis phase, and this we call it as trans phase. From trans phase, that transport vesicle will be released that joins with the plasma membrane, and that material will be released outside. So that's how the functioning of Golgi apparatus as well as endoplasmic reticulum takes place. So now we are supposed to take about the structure of lysosome, types of lysosome and their function. Then later we'll take about the vacuole. So if you take lysosomes, even these are single membrane bound cell organelle produced from Golgi apparatus. It contains different types of hydrolytic enzymes. These hydrolytic enzymes can break down proteins, lipids, carbohydrates, even nucleic acids can be broken down. All these enzymes are active at acidic pH. Such type of enzymes which break down proteins, lipids as well as carbohydrates, they are called lipases, proteases and carbohydrases. All these are the hydrolysis enzymes. These enzymes are capable of digesting carbohydrates, proteins, lipids as well as nucleic acids. But if you take the types of lysosome, we can classify the lysosome into 4 types. But here I have given 3 types: primary lysosome, secondary lysosome, autophagosome. If you take the primary lysosome, you can see in the on the screen there is the Golgi apparatus. The lysosomes are produced. Whatever the lysosome that is produced, the newly formed lysosome, that we call it as primary lysosome. And later during endocytosis, it's a type of phagocytosis. If the solid food is taken, then it is called phagocytosis. If the liquid molecules are taken, then we call it as pinocytosis. Then the vacuole that is formed after phagocytosis, that we call it as phagosome. Or if it contains liquid molecules, then we call it as spinosomes. Such type of phagosome joins with primary lysosome, then the resulting the lysosome, that what we call it as secondary lysosome. So secondary lysosome, it is the lysosome which has joined with phagosome. Then digestion takes place inside this the secondary lysosome. After digestion, the byproduct will be present inside the lysosome. Such type of lysosome which contains byproduct, they are called residual lysosomes. Later, these residual lysosome during exocytosis that will be sent outside. The waste product will be sent outside. So the newly formed lysosome is called primary lysosome. The lysosome that has joined with the phagosome, that what we call it as a secondary lysosome. The secondary lysosome involved in digestion. After digestion, the waste products are produced. Whatever the waste products that are present in the lysosome, such type of lysosome, we call them as residual lysosome. During exocytosis, it will be sent outside. In the same way, if you take autophagosomes, the lysosome that are released from Golgi apparatus, this will be the Golgi apparatus and this will be the lysosome. The lysosome, they take up old cell organelles. Such type of lysosome which takes up old cell organelle and digests them, we call them as phagosomes, autophagosomes. So they digest or remove the older cell organelles. That process what we call it as autophagy. So these are the different types of lysosome. The newly formed lysosome, it is primary lysosome. The lysosome that has joined with phagosome, that what we call it as secondary lysosome. Then the lysosome which contains waste products, that we call it as residual lysosome. Then later the lysosome which remove the older cell organelle, that we call it as autophagosome. So these are all different types of lysosomes. So lysosomes actually produced from Golgi apparatus. Then coming to the next one, if you take the vacuole, the next one, vacuole. Vacuole is also a single membrane bound cell organelle that is mainly found in the plant cell, but also found in animal cells. So what does it contain? It contains water as well as sap as well as excretory product and also certain material that cannot be eliminated. Such type of materials will be present inside the vacuole. For example, there is the plant cell and here you find the nucleus. Then if you take the vacuole in plant cell occupies about 90% of the cell. So it is a single membrane bound organelle. That membrane that surrounds the vacuole, that what we call it as tonoplast. So the vacuole contains, it contains water, it contains sap, all the nutrients are present. Then excretory product, it contains excretory product. If you take in plant cell, this vacuole represents about 90% of the plant cell volume. This tonoplast allows the molecules to move from a region of higher concentration to a region of lower concentration, that is against the concentration gradient. So it is an active process, so that the mineral concentration will be more when compared to the cytoplasm. It is mainly because of the characteristic of tonoplast. Tonoplast take part in active transport. It makes the minerals as well as the ions to pass through the tonoplast, and all these minerals as well as the ions will enter into the vacuole by the active transport. It is going to utilize the energy. So that's why the minerals as well as ions are more in this vacuole and compared to the cytoplasm. Then if you take the animal cell, if you take in amoeba belongs to protozoa. In protozoans, the best example for protozoan, one will be the paramecium, the other one will be the amoeba. The amoeba, it contains the nucleus. Even paramecium contains two types of nucleus. One is macronucleus, micronucleus, another one. But along with these cell organelles, the protozoans, particularly amoeba as well as paramecium, they contain the vacuole. They contain vacuole. These vacuoles are mainly meant to control the water concentration inside the organism. Maintenance of water concentration, we call it as osmoregulation, and also eliminate the waste product from the organism. Even in paramecium, you find such type of vacuoles. These vacuoles are called contractile vacuoles. They are called contractile vacuoles found in amoeba as well as paramecium, that is particularly in protozoans. Even in protista, even protozoans belongs to protista. Even algae belongs to protista. In protista, you find vacuoles, but those vacuoles are not the larger vacuole which are not involved in osmoregulation, but they are nothing but food vacuoles. So in protista, you find food vacuoles. In amoeba, you find contractile vacuoles. Whereas in plant cell, the largest vacuole will be present. It represents about 90% of the plant cell. Most of the waste that cannot be eliminated, that will be stored in the vacuole. If you take the major function of this vacuole, it creates hydrostatic pressure, it provides a mechanical support to the plant cell, it maintains the shape of the cell. That is the functioning of the vacuole. And also it stores all the waste that cannot be eliminated by the plant cell. So here I have given a few extra information. Vacuoles store many toxic compounds of the cell, including byproducts of the metabolic pathway. In animals, you find excretory system to eliminate a waste product, but in plants you don't find such type of system. So the plants cannot eliminate the waste. That's why the such type of waste as well as minerals will be stored inside the vacuole. When the minerals as well as the waste products enter into the vacuole, automatically water will enter, because the concentration of these ions as well as minerals will be more in the vacuole. When water enters into the vacuole, then its hydrostatic pressure increases. That hydrostatic pressure provides a mechanical support to the plant cell. So this is all the information you are supposed to remember for board exam for lysosome as well as vacuole. So for lysosome, what you are supposed to remember for board exam, it's a single membrane bound cell organelle. It is produced from the Golgi apparatus. It contains a large number of hydrolytic enzymes. All those enzymes are hydrolases. You find lipases, you find proteases as well as carbohydrates. All these enzymes have the capacity to break down proteins, lipids as well as carbohydrates, even nucleic acids. But these lysosomes, they are commonly called the suicidal bags in a cell, because if these lysosomes, if they are ruptured, they can destroy the cell in which they are present. That's why they are called suicidal bags. And if you take vacuole, what you are supposed to remember? In vacuole, you are supposed to remember about the membrane that surrounds the vacuole. It is tonoplast. So what it contains water, sap as well as excreted product, and also certain waste that cannot be eliminated by the plant cell. Such type of vacuoles are found in plants as well as in animals. In some animals, particularly in amoeba, you find vacuoles which are mainly meant for osmoregulation as well as excretion. But protista, you find the food vacuoles. But if you take in plant cell, it is one of the important characteristic of plant cell. 90% of the plant cell is represented by the vacuole. The tonoplast allows the molecules to pass into the sap that is present in vacuole through the by using energy, so that the mineral concentration as well as ion concentration will be more inside the vacuole and compared to the cytoplasm. So this creates the hydrostatic pressure that provides mechanical support to the plant cell. So these are the information what you are supposed to remember for the board exam. But if you take some important information required for competitive exams, so that I have taken in MCQs. In plant cells, the byproducts of the metabolic pathway are stored in. So all these byproducts have been stored in vacuole. So the answer should be the first one. They are stored in vacuole. But what is the functioning of mitochondrion? Mitochondrion, it is a singular form of mitochondria. Plural form, we call it as mitochondria. Singular, we call it as mitochondrion. Mitochondria, it is involved in releasing of energy. That's why it is called powerhouse of cell. Nucleus, it is a double membrane bound cell organelle. It contains nucleic acid, that is a genetic material. Chloroplast is involved in photosynthesis. So all the byproducts in plants, they are stored in vacuole, because plants do not have the excretory system. Then coming to the second one, a tonoplast is the membrane that bounds the vacuole. So that the membrane that covers the vacuole is a tonoplast. It contains many dash transport system. It contains active transport system that pumps ions into the vacuole. Against the concentration gradient, ions are pumped to enter into the vacuole. Such type of process requires energy. Any process or any transportation that requires energy, that we call it as active transportation. So the right answer will be the first one, active transport. Due to the high concentration of ions in the vacuole, water enters by, in what way the water enters into the vacuole? It is by osmosis. It is not by diffusion. Diffusion of water itself is called osmosis. So that if you take the B, the pressure is developed. It is not electrostatic pressure. It is actually the hydrostatic pressure. And water enters into this vacuole by osmosis and not by diffusion, and also not by active transport. Minerals as well as ions will enter into the vacuole by active transport. water enters by osmosis. So this creates hydrostatic pressure. So that C is wrong. Even D, it is not facilitated diffusion. Facilitated diffusion, the molecules are transported through certain proteins, the carrier proteins. Along the concentration gradient, such type of carrier proteins helps in facilitated diffusion. So the right answer for this one will be B, osmosis and hydrostatic pressure. Then if you take the similar information, the fourth one, similar to lysosome present in animal cells, plants contain dash for intracellular digestion. So the plants contain what type of the structures for intracellular digestion? They contain secondary lysosome. For intracellular digestion, the right answer will be A. What of the following molecule is not transported through the secretory pathway of endomembrane system? So in endomembrane system, lipids have been transported, even polysaccharides have been transported, even proteins are transported, but not nucleic acids in endomembrane system. So the cell organelles that are involved in endomembrane system, we have already studied that: endoplasmic reticulum, Golgi apparatus, lysosome as well as vacuole. So here the right answer will be nucleic acid. Cell organelle involved in endomembrane system. Mitochondria is not involved in endomembrane system. Mitochondria is a double membrane bound cell organelle. Chloroplast is also a double membrane bound cell organelle, even peroxisome. But the vacuole, it is single membrane bound cell organelle. It is actually involved in endomembrane system. So the answer will be D. So all these are the organelles, whatever we have studied, they are involved in endomembrane system. So if you take the previous video information, I have discussed about endomembrane system, that is cell organelles in which their function has been coordinated together, what we call it as endomembrane system. The cell organelles which are involved, one will be the endoplasmic reticulum, Golgi apparatus, the another one will be the lysosome and the vacuole. And also I've discussed it few MCQs. If you have understood, please press the like button. I'll take another cell organelle in the next video. Okay then, bye.